The Australian Grains Gene Bank at Horsham in Victoria has germplasm collected from farmers, roadsides and breeding programs all over the world. It's a vital resource for plant breeders. Finding new sources of plant genetics takes really dedicated seed collection work, and there's none more dedicated than the curator of the AGG collection, Dr. Bob Redden. His recent work with peas in China has uncovered sources of genetic diversity never before included in plant breeding programs. And thanks to a GRDC collaboration, Australian plant breeders will be the first anywhere in the world to have access to them.
Many agricultural crops originated in what's called the Fertile Crescent in the Middle East. From this genetic motherlode, cereal and pulse crops were dispersed around the world. However, in countries like China, some of the genetic material remained isolated for centuries and is only now being accessed by international crop breeding programs. As Bob explained to me, field peas are a good example.
The usual supposition is that some of the most diverse material will be found near the centre of origin where it was domesticated. And when it is transferred, it migrates with people around the different continents, there's usually a narrowing of genetic diversity. But what we found was wider genetic diversity in the material from China than in the Middle East. It confounded our expectations. And we don't have any simple answer for this, except that there's a huge array of diverse environments. They were isolated from Western Europe for a very long time, nearly 2,000 years. And so these crops continued to evolve in isolation in China. And, so it's considered to be sort of a whole range of selection pressures, new mutations, and rearrangements in the germplasm, possibly more introgression from a wild relative up around here in Central Asia, because they add in diversity. And after domestication, crops were not completely isolated from the wild material. They were often grown in the same general localities, and the wild material was sometimes even a weed in the field of domesticated crops. And so consequently there could have been the occasional exchange of genetic material.
Samples of these Chinese-sourced field peas now held at the Australian Grains Gene Bank show the diversity of the collection. Different colours, size and shape are common and are typical of the mixed bag that was crop production in ages past.
The idea of having a very pure, homogeneous variety has only come with modern food processing and modern plant breeding. So prior to that, material typically would be a landrace like this, a mixture of types, speckled, plain, different colours. And this was all what the farmers normally grew. And it was probably good practice, which the farmers knew about, because some years would favour some parts of the mixture and other years some other. So they got something which was reliable to give a stable return. So they went for stability and guaranteed return, and when new modern varieties came around, there was then the option to go for the highest return, particularly when you add inputs like fertilisers, etc.
Sourced from across China, the field pea germplasm are of enormous value to Australian pulse crop breeders.
This material is genetically isolated and has not been used for plant breeding in Australia. So for us, it's new material. Most of our material is based on Middle Eastern European genetic material. So there's a real possibility we can get new and different types of genetic resistance, yield expression genes out of this Chinese germplasm. So that's part of the value of what we're doing. And as I said, we had when we evaluated a cross-section from Yunnan across to Harbin, a cross-section of peas from China, and we planted them all together and watered them with salt after they germinated. These two stood out as being very resistant to salt. They kept on growing almost normal. So that's really interesting. And these have been used in our plant breeding program here in Australia to breed a higher level of salt tolerance in our commercial pea varieties. So we're transferring the genes from these into our commercial varieties, like this one here, Casper, which is a sort of semi-dwarf, erect variety, semi-leafless, with little vines on it that bind the plants together as they grow. So it's essentially non-lodging. So a very good agronomic variety. But there's always room for improvement, and things like adding salt tolerance to this kind of variety is the way we're going.
As a result of this kind of quite privileged access to material we've never used before in Australia, and probably plant breeders
everywhere
have never used
outside
before.
of China, yes.
And that combined with the new Australian Grains Genebank will provide a more streamlined access for this never before used material into Australian plant breeding programs.
Correct, it really will, because the new Genebank, as Sally will tell you, is going to have a new database which will be online, which will have all our inventories listed for peas, lentils, chickpeas, wheat, maize, all of it. It'll be available online with a shopping cart where you can tick off which ones you want. We haven't had that facility before. We haven't had a one-stop shop before. And it will improve and facilitate access for students at universities, for plant breeders, for pre-breeders, and people overseas. And the exchange of material with overseas Genebank will always be important because all of our crops are imported, except macadamia, and that's not a grain crop. But all of our grain crops are imported, and the germplasm that supports the plant breeding of those crops also has to be imported. In a few cases we have distantly related indigenous wild relatives which can also be useful, but within the domestic gene pool we have to look overseas, and hence if we can access the ancient variation in village varieties from around the world, that's a gold mine for us.